All right. So thanks, Andy. Great initiative, worth learning. And now to the intricacies of making beer. I have known Ken Caswell for a relatively short time, but once he and another friend of mine, Professor Joe Askren, who was our first speaker in my Rotary year, told me, why don't you come and attend a rather unique class? And when we talk about a class like Dr. Ladwig over here or I, we are talking about serious stuff like Python or C++ or IT security. And we are generally struggling to get our students to come and attend our class. I found the exact opposite. Students are highly enthusiastic. Students have got their writing pads and they are also ready to do practical. So I thought this will be a fantastic speaker to invite, and I invited Ken Caswell. He's a professor, he's a teacher, instructor in College of Hospitality and, okay, what is it? College of
Science and Mathematics.
College of Science and Mathematics. I thought about
hospitality is close.
Hospitality is very close.
There's a good reason he's got this confused. I'll
But
explain that.
I can summarize by just one word that he's now a very, very dear friend of mine for more reasons than when we work together in some committees. We do a fair bit of work in the service side of the university. And we also have cross-discipline, we are encouraged to set up cross-discipline projects, and that's what Ken and I are working on. So with that small introduction, please join me in welcoming Ken Caswell. So you've got your 20 minutes and
Thank you. Thank you for having me. I haven't been up this early since my days in the military. And they started that stuff too early, so I had to get out. No, I put in my 14 years. So how many of you actually have some form of chemistry or biology background? I've got my work cut out for me. Like most of my students who join in general chemistry anyway, they tend to lack the, oh, I'm sorry.
No, you can walk around, I'll move.
I tend to be a bit of a dynamic classroom person, so. Anyway, so the brewing process itself is insanely complex from the chemistry and microbiology standpoint, and even from physics and so on and so forth. So we could spend years talking about just water chemistry. Charlie Bamforth out at UC Davis has actually spent about 30 or so years studying just that, the water for brewing, and also foam were the head on a beer. We're going to focus a little bit more on yeast because they're the ones that do all the really cool stuff. Before we get there, though, I want to address the Joe Askren College of Hospitality, Ken Caswell College of Science and Mathematics situation. So Joe Askren is the gentleman on the left here. This was at the Sip the Sunshine event that was last fall, which was actually a big craft beer festival that was put together by a member of the community to feature craft beer, but also our budding Science of Beer program. Matt, let me clarify that, it's not a program. Science of Beer classes, and there are two of them. There is the College of Hospitality's Intro to Beer Science, which is Joe Askren's section, and then mine is the Chemistry and Microbiology Beer class. We teach them at the same time, but we do have a little bit of slightly different angle between the two of them. My course is a biology upper level elective, and Joe's is open more broadly to non-science majors. The point of bringing this up is that we wanted, from the very first day I taught general chemistry, I realized that, nobody wants to learn general chemistry. Indeed, if I go to a dinner party or a cocktail party and I mention that I'm a chemist, I blink and there's usually nobody standing in front of me anymore. Which is really a big surprise that Bhuvan wanted me to come here and give a talk. Anyway, Joe and I got an opportunity to, long story, but we got an opportunity to pursue more experiential learning opportunities. So rather than having four white walls and just talking about the chemical equations associated with beer and brewing, and, well, just science in the day of a life, we decided we were going to bridge out and work directly with industry to give students that opportunity of boots on the ground, that nuanced stuff you can't get from a textbook. And I've learned a lot in that process from industry as well. So we teach the science of beer classes. I am also teaching a science of cooking class in the spring. Joe is a chef and also an instructor. He just got his PhD in education, is that correct?
Yes.
And so we pursue very unique approaches to getting education across. We also have an international food and beverage science and culture program that we teach over in Tuscany and in Lyon at Institut Paul Bocuse and a few other really prestigious institutions. Our ultimate goal is to have a fermentation food and beverage science program. We'll see how that works out. We have momentum. Okay, so there are 4 main ingredients. How many of you actually beer drinkers? All right, good. Normally I'm speaking to a group of wine drinkers and they can't appreciate beer. My comment there is you actually haven't had beer yet. But so the 4, regardless of what style it is, typically there are 4 main ingredients. There's water, as I mentioned. There's a huge amount of detail involved with water alone. You have to have the right ions in water. You have to have some right magnesium and calcium. You have to have some zinc in there. Primarily to get some of the processes we're going to be talking about happening with yeast. They need these ions to get their enzymes and such going on. You need the grain. The grain is where we're going to get our sugars, our food for the yeast. We're going to feed the yeast. What we ultimately want is the yeast poop. That is the ethanol. That is the alcohol and the carbon dioxide. The hops have a relatively new addition to beer as far as the overall brewing process. We've been brewing either by accident or by intention for thousands of years, but hops only have been around for hundreds of years. We've been adding things like herbs and that sort of thing to kind of balance out the sweetness of beer to make it more palatable, but also to add some medicinal value. And it turns out hops have some amazing medicinal properties to them, and of course, like anything in the right doses. We're actually just unveiling some of these, and this is probably this is definitely a time for another talk, and talking about how they even have anti-cancer and all kinds of other really cool compounds in there. And we are largely unknown the total number of compounds that are in hops yet. Yeast is where we're going to focus today. Then there are what we call adjuncts. Adjuncts are the additional things that can be added to beer. They're the kind of peripheral things to add it, some unique flavor, unique aroma, unique things like coriander or coriander and some other oats and that sort of thing. And then brewing with other bugs. Typically we use yeast, particular varieties of yeast, particular strains of yeast. And there are lots of those. And they're very specific to a style of beer. We'll talk a little bit about that in a few moments. The other bugs, though, have traditionally been the things we were trying to keep out of beer. Mm-hmm. And indeed, things like Brettanomyces, which tend to give more of a souring effect. But now the really cool approach is to try to get these bugs back into the beer to add that really interesting dimension to it. Just if you've not been to Coppertail Brew, go to all the craft brewers. They're all amazing for a lot of different reasons. Coppertail's got a particularly interesting artist from Oregon who does a lot of their artwork, and this is one example of it. Their artwork on the lab. So the great variety of beer that have spawned relatively recently, which is an expansion based on recovering from a contraction in beer brewing, that being centered around prohibition and such. Food, when we're talking about the flavor, taste, aroma, and mouthfeel of beer, they come from a lot of chemical compounds. The estimations of about 800 compounds associated with flavor in beer, and about 600 of them probably come from yeast alone. Cool. And that comes from just their normal life cycle. And we can go into a lot of detail as far as that goes. We can speak in terms of their genetics, in other words, their DNA, deoxyribonucleic acids, their blueprint, and what they need to be a cell itself, and to the metabolism that they need to do the things they need to do to create a colony, to create other organisms. So they need all these individual compounds, and within those individual compounds, we've got all kinds of other compounds that are either derived from nucleic acids or proteins and that sort of thing, but they also are parts of things like hops and such. So what we're focusing on is creating an environment to make yeast happy. We're facilitating their home, if you will. The goal is to get a lot of this stuff, sugar. Glucose, in particular, is what they go after first and foremost. They will metabolize other sugars, but they will go after only those other sugars after they've taken care of the glucose. We're probably most familiar, and this is the structure of a glucose molecule. It can be in this open form or it can be in this closed form. That really isn't that important to us right now, but what is important to us right now is that it has 6 carbons in it. Okay, this is going to become important in the well. Those are equations. We have 1, 2, 3, 4, 5, 6 carbons, and ethanol is just 2 carbons with an OH group. An OH group is one of these OH groups off to the side. What we end up doing, or what yeast end up doing, is they end up metabolizing, like we take food and put it into our bodies, right? Our body goes through a whole series of processes to break that food down so that we have the energy to stay upright and listen to somebody talk about chemistry. It's 7, 8, where are we, 8 o'clock in the morning? Right. And what they're aiming at here is a compound called adenosine triphosphate, ATP. It's produced in what's called a mitochondria. It's one of the smaller organelles within a cell. And so what they're doing is they're breaking this compound down to get energy. We do the same thing, but in a little bit different approach. Our brain largely feeds on glucose. It doesn't matter what crap you put in your body, it's aiming for the glucose. So yeast, what we're often getting a lot of what's called sucrose. Sucrose is a glucose molecule and a fructose molecule that are fused together in what's called a glycosidic bond. Starches are long chains of just glucose. They're just beat up, just one chain onto another. With me?
Yes.
Okay, good. So amylose and amylopectin are the 2 major starches. And for brewing, or for beer, we use all kinds of different grains, but the primary grain is barley. And we use barley because it has this unique ratio of the starch, these sugars, and protein. But what you need to really know here is that amylose and amylopectin have, again, the same pairing up, or this glycosidic bond between individual glucose molecules, but they are different in the sense that there's branching in the amylopectin, which means that there are different enzymes, different proteins that break these things apart, because the yeast can't use these by themselves. They're too big, they're too clunky, and what they need actually is to break these things down into individual glucose molecules, 1 ring structure, or 2, which is called maltose, or 3, maltotriose, and so on and so forth. So there are different chain lengths that can be used for food by the yeast, and then there are different chain lengths or different branching that give you that mouthfeel. They're what are called dextrins. They help actually with, well, just that, to give it that sort of body, that fullness of a beer, or lack of it. And then we need proteins. The proteins help create the foam or the head structure on beer so that you get that nice frothy foaminess to it, which most people, when you go to pour a beer, they complain that they don't want all that because they're not getting as much beer, but you need that foam, you need that head on that beer to help bring those aroma compounds to the nose, because most of flavor is through the aroma part of it, not so much the taste part. It's actually in a much deeper seated part of the brain. So really quickly, we're going to go through the brewing process, and I'm not going to really focus on any one aspect of the brewing process. We're going to focus on what the yeast are doing. So the first thing we need to do is get our barley in the way that we need our barley. So the first step of this is what's called malting process. And the malting process, what you do is you take these grains, and these grains are of course just baby barley. And in the malting process, the first thing we do is we start them growing. And I say we, the brewers actually usually don't do this. This is done before the brewers get the grains. What they do is they start them growing. They create conditions, they add some water to them to get the seed, the baby going. And growth is a destructive process. It's a chaotic process. Think about this. You've got a seed. You have to break that outer wall of the seed so that we have to get the nutrients for that embryo to start growing. And that's what's going on in the malting process. The kilning process is to stop that process. So the point of this is to get them going so that you start creating a little, what's called a chit, which is just the little rootlets start to come out the backside of the grain here. You start to break down those longer starches into some smaller sugars. And then we want to kiln it, or in other words, dry it to stop that process. And what we want to be careful of in that is that we want to preserve those very important enzymes, these proteins that'll break these sugars down further when we want to do this in the brewing process. Still with me? Yeah. And in the kilning process, we can also do these, we can go through a series of reactions called caramelization and the Maillard reactions. And in those, we end up getting this unique color and flavor profiles out of the grain itself. In other words, you can get much more of a coffee or chocolate vibe from a grain by toasting it longer. We know this with literally with coffee, right? You can get a coffee that is not as roasted as long. It's going to be more acidic and have maybe some more floral and such vibe to it. But as you roast it more, it becomes less acidic, more that sort of like chocolatey and heavy. Well, what most people would perceive as a heavier vibe to it. All right, so we've got the grains and we've got them at least where we want to start in the brewing process. Oops. And then in that, we will go through what's called mashing. And that's just taking the grains and putting them in the right temperature water to release the right enzymes. Now, there are actually a series of enzymes that do this. And they are depending on whether we need them to take individual sugar, smaller groups of those sugar rings off of the larger starches at the ends, or whether we want to aim for them at the branching points. Because one enzyme can't do the other work. Does that make sense? Mm-hmm. Yep. And again, the smaller sugars are what the yeast can deal with. The bigger branching sugars are going to give us that mouthfeel. Right? So if we hit it at the wrong temperature, we may not end up with enough of the free sugars so that we can't feed the yeast, and we don't end up with ethanol, and you got sort of a lower alcohol content beer, which may be what you're aiming for, but then it's going to have a bigger mouthfeel to it. So it's going to be more of a maltier flavor to it. You can dry out a beer by literally just turning as much of that starch into sugars that the yeast can consume, and then let the yeast do their job and just completely take all the sugars out of the process. After that, we go through what's called sparging and that sort of thing. That's just washing the sugars away. Not too exciting and interesting. And then we put them into what's called a kettle boil. That's literally that. It's put into a kettle where we boil that. And in that boiling process, we do various things that I'll point out here in a few seconds. All kinds of really cool chemical reactions are happening in that case. And that's usually where we add the hops. And we actually, depending on the style of beer, you'll add the hops in different fractions. If you add the hops very early on, you'll end up with those bigger, bitter type beers like IPAs and such. The Americans have taken the bittering process to extremes in adding lots and lots of hops throughout that whole boiling process. If you want much more of the aroma compounds, the smell, those that are really give you those delicate and interesting smells that contribute to overall flavor, you want to add those later in that boiling process. But again, today's talk's not really so much about hops, which are cool and interesting. Yes.
Is all that happening in those big dens?
Yeah, absolutely. So there are actually different types of, there are actually different types of vessels within a brewery. You have, and historically they have been everything from big concrete open, like pools, to being in wooden, much more like giant wine barrel type things. Into modern day, much more in the stainless steel and even what we call cylindrical. In other words, they're cylindrical at the top, and then they end up into sort of a little conical, inverted conical, that allows the draining of the yeast or the settling of the yeast into the bottom there and separate.
So that they can heat with that, they can do everything they want.
Absolutely. And you are transferring from one type of vessel to another in the brew house. In some of them, they can combine some of those steps within a particular vessel. But typically, you're moving from the mashing to what's called the lauter tun.
Okay, so you move it to another place.
To the fermentation.
Okay.
The fermenters tend to be the conicals.
When do you add the yeast?
You add the yeast at the very end. So all of this most of brewing actually is sanitation and cleaning. It's a lot of hard work in making sure that you keep all the other microbes. And we're really attending to these little critters. We're creating a very, very happy, sugary environment for they to live. We're eliminating all of their competitors, and we're going to talk about some of the things that they have evolved to do to eliminate their competitors as well. But yeah, what we're doing is we're creating their happy little home, and then we let them do their thing.
When you add the hops, is that when you would add the flavors? I drink White Chocolate blueberry beer in the summer, and it's blueberry beer.
Yeah.
Brewed by White Chocolate Brewing Company, and it's a wonderful craft brewer in Massachusetts. When would they put the blueberries in?
Typically, we add, for instance, we just did what's called a milkshake IPA, which had peach and vanilla bean in it. We added those at the very end with the yeast. So we add it after all that process.
After the hops.
After the hops, typically. You can add fruit earlier on in the boil, but then you're risking going through chemical reactions that you don't necessarily want to. When you're boiling something, one of the reasons we add the hops during the boil is to do what's called an isomerization process. In other words, we're changing a molecule slightly, and one of the components that are in hops, we change their structure a little bit through the boil because you're adding energy by boiling it, right? And changing that structure makes it more soluble in a water environment, but also changes the flavor and aroma profile of it as well. So typically we add the fruit, usually what's called, well, we can add hops too in that fermentation when we add the yeast at the end. That's called dry hopping. That's after the boil. And then we can also add fruit and all kinds of other things. Some of the other adjuncts we would add during the boil, for instance, I'll add coriander or bird of paradise or something like that in the boil process. Go ahead.
Just to answer, in Maine, they put the blueberries in the glass. Then you just put some bugs on top, and they say that's the blueberry.
Can you tell us how long these different processes last?
Sure, absolutely. So in the mashing process, what you're doing is you're putting those grains into a container, and then you're adding the right temperature water, and you usually let that sit for about 90 minutes or so. And in that process, the enzymes are starting to break down those starches into smaller sugars that the yeast can digest. There's what's called Alpha-Amylase and Beta-Amylase, and we'll talk a little bit more about both of those. The sparging process usually takes, you want to be careful. What we have to do is stop those enzymes in that mashing process. So we want to remove those proteins from the process. And in that, that's part of that sparging process. We go through, we add a high enough temperature where we end up denaturing these proteins. In other words, we're kind of killing the proteins. They're not really alive, but we're just destroying their capacity to do this work anymore. And then we're going to wash the grains with some additional hot water. And by adding some additional water, of course, we're diluting this sugary environment. And that just takes a while to wash out all those grains. Then you're going into the boil. The boil is usually 60 to 90 minutes. You don't want to boil too long because you don't want to take the chemical reactions too far, but you need to have a long enough period of time where we can go through these isomerization and other processes. And then the fermentation process, after the boil, we've cooled it down and we've added the yeast. That depends on the style of beer. Lagers tend to take a little bit longer because they're done at a cooler temperature and a different type of yeast. Can take months, but usually we can knock that out in 3 to 4 weeks. Whereas an ale, pretty easily within 2 weeks, they're done converting the sugars to alcohol. And then there's a whole process called conditioning. Not all beer needs to go through a conditioning process, but most green beer, green meaning that it could use a little bit of conditioning, goes through some additional chemical reactions by sitting either in a cask. These days we're doing a lot of really interesting things by taking old whiskey or even wine ports and that sort of thing and putting the beer in there and then extracting some of the cool compounds out of that.
Is there a real different process when you're in the conical boil? We were over in Europe and went to a winery and they had an egg-shaped cask that they put this wine, it was a special wine, and they said that when they put it in there, the whole thing was sealed like an egg and smaller at the bottom, but they said that when it ferments, it goes in a swirl inside this egg and ferments inside there, and it's a unique process that
He's not been listening, we're talking beer, and he's talking wine.
I know, but he's
just
talking about
kidding.
the conical versus the vertical, and I'm wondering if that makes a big difference in it, or you've seen any big difference that way.
So the conicals the conicals have that just that is a pointed, is like a dunce cap turned upside down, right? So it's in the bottom. You've got the cylindrical part, and then so one of the reasons a couple of the reasons they've gone to this type of setup is, one, they can get more happening in a vertical space rather than having to deal with a horizontal space, right? So in the open, historically the big open concrete vats, you need to have a lot of surface area, right? If this were our floor of our brewery, if we were taking up this entire space with this big horizontal space, you wouldn't have space to do other things, right? So now we can go vertical with this thing. Now there's some issues with that, some very physical issues with that, but the conical itself offers this sort of, look, they're producing a lot of CO2. You're talking about a lot of yeast that are converting a lot of sugar, and there's a lot of energy released in this process, so we have to put cool jackets on these things, so some glycol on there to cool it down. But in that process, with that heat and that sort of bubbling, if you will, the CO2, it does a fair amount of circulating on its own. As we're going to talk about here, hopefully in a few seconds, is that yeast actually don't need a lot of oxygen. Matter of fact, they really don't. We actually don't want them to have a lot of oxygen because we want them to go in a particular pathway, which they're normally prone to as long as we have the sugar content in the right way. So yeah, in both processes, wine and beer, we are talking about yeast, and we're talking about a very similar fermentation process. There are unique aspects of both the wine and the brewing process. I haven't seen the egg, but usually when we're talking,
there's like 5 of them in the world.
Yeah, there are all kinds of really cool, innovative engineering that is happening within the brewing industry now as it's really exploding, and wine, of course, has been kicking along for quite some time. But one of the cool places for a whirlpool is actually at the end of the boil part of this, where we want to get rid of some of these, the residual hops, and we want to get rid of some of these residual proteins through what's called the hop break and that sort of thing. But the conical...
Out of time.
We're out of time? I haven't even started.
So a couple of minutes, so maybe you can wind up, please.
All right. Well, wait, we didn't actually get a chance to talk about any of the yeast. Let's see here. Oops.
Well, that means there's no exam at the end.
Right. All right. So what we would have the different yeast trying to pull off these individual sugars, and there are those branching points. So the different types of enzymes will do that. This is the enzyme that will grab a hold of the starch and break it in very unique places. The goal is to create this molecule. This is ethanol. Ethanol is the same molecule, whether it's in beer, wine, distilled spirits, whatever. That's the thing that gets you drunk. All of
gas?
What's that?
Ethanol, same, not
Petrol.
gasoline. I mean, ethanol, you hear ethanol in your tanks, all that stuff.
another way that they can get corn products into something else,
All kinds
but
of gas.
Ethanol is the thing that gets you drunk. Carbon dioxide is so we produce one of these and two of these for every one of these. So why, real quickly, why, we'll skip the boil here for a few, because we run out of time here, but the point of yeast here is that normally if they had we go through what's called respiration. We take one of those sugar molecules and we break it down completely. We go through this process where we take sugar and break it down completely into just carbon dioxide molecules. And we tap that thing out for all the energy it's worth. Whereas yeast have done this really unique thing. They break it down only part of the way. They break it down to just the ethanol and just a couple molecules of this carbon dioxide. This created a few interesting advantages for them. One, most organisms don't like ethanol. It's antiseptic. In other words, their poop is deadly to everything else. Now, it's a slippery slope here because they can only survive in an ethanol environment up to a certain concentration as well. They also are able to, and so what they do is they sacrifice the amount of energy they're going to get by not breaking down the molecule completely as a matter of expediency as opposed to efficiency. In other words, not getting maximum amount of energy, but you're making a safer environment in which you can survive in.
Yeah.
It's over. Just one little question. What's the major reason for a bad batch?
Usually it's light struck. It's usually after the beer that's been made. The worst enemy of beer is light. Light, because it's energy and it converts. You can get light struck beer in a clear glass bottle like that.
With your extensive knowledge of brewing and beer, do you have a favorite beer that you?
I like Belgian tripels, actually. I like the big Belgian ales. They tend to be higher in alcohol content. They tend to have interesting flavors to them.
That's good.
All right, Ken, please stand here. That was most intense on a Black Friday, but I had invited Ken to talk on the intricacies of chemistry in making of beer, and he did a great justice. Yes, Jim.
You promised us samples.
I have all the samples for you, but this is an Amish restaurant, and we follow the 4-way test. Is it the truth? We can't have a sample in an Amish restaurant. Did I do that well?
Sunday, samples are
Why
at Sunday at the
we
Cricket.
had mimosas for the.
We never have. Okay.
I did bring some hops and some grains if you want to eat those.
Okay, thank you. So on behalf, no, please stay here. On behalf of our entire club, I would like to present Ken with a coffee mug in which he can experiment tasting of beer, and we can all join him. So once again, please join me in thanking Ken. Thank you very much. We have $4, and the last 3 digits are 710.
710.
This thing is rigged.
710. Who worked the front desk?
710.
Worked the front desk?
Next week we have the district governor elect coming, and we have a new member induction. Please make sure you all come and celebrate. Thank you. See you next Friday. And anything you can.